Source and recovery of DNA. The source of the DNA would be blood, semen, saliva, urine, hair roots, teeth, bone, as well as tissues. We can purify the DNA from all these cells or the tissues. These are the source of DNA. That can be blood, that can be the teeth, that can be the sperms, that can be the hair roots, certain things by which we drink or eat, the bone and the tissues. So these are all source of DNA. These are all the cells or the tissues from which we can purify the DNA. So in addition to this, there are also source of DNA in the form of cigarette butts, envelope and stamps, fingertail, chewing gum, bite marks, as well as feces. We can also purify DNA from these things with the help of certain techniques or by using certain kits which has been designed to purify the DNA from these things. The main source of DNA is the blood. That can be the human blood or blood of the animals. So that is the best thing from which we can easily purify the DNA. So the blood consists of three kinds of the cells. They are red blood cells, white blood cells, and platelets. Red blood cells. Red blood cells, they are also called as erythrocytes. There is no nucleus in the red blood cells. It means if there is no nucleus, then there is no DNA. So we can say that there is no DNA in red blood cells. So they are not useful for the DNA section or for the purification of the DNA. While on the other hand, white blood cells, they are also called as leukocytes. White blood cells, they have nucleus. Nucleus is present within the white blood cells. If there is a nucleus, it means there are chromosomes or DNA. DNA is present within the white blood cells. So we can purify DNA from the white blood cells. These are the blood cells. Blood cells are of three types: red blood cells, platelets, and white blood cells. So out of these three types of the cells of the blood, only the white blood cell are those cells from which we can purify the DNA, because the DNA is present in the white blood cells. They have the nucleus, and within the nucleus there is DNA. So nucleus is present in the white blood cells. If we see that how much amount of DNA is present within the cells, so in a diploid cell that has 2n chromosome numbers that contain approximately 6 picogram of the DNA. So a diploid cell has 6 picogram DNA. While on the other hand, a sperm, which is haploid, that contains approximately 3 picogram of the DNA. Theoretically, we can recover the DNA from 1 microliter blood is 30 to 60 nanogram. From each of 1 microliter blood sample, we can purify 30 to 60 nanogram of the DNA. It depends on the technique that we have used. If we purify the DNA by manual procedures, the amount of the DNA would be less. But if we use certain kits which are sensitive, we will get maximum amount of the DNA from 1 microliter of blood sample.